us all sa ating 25th Kapihang OH. Ladies and gentlemen, our PCOM Quezon City Chapter President, Dr. Catherine Artais Carriaga.
Maraming salamat, Dr. Margo. Magandang gabi po sa inyong lahat na aming tagapanood at tagapagsubaybay mula sa iba't ibang lugar ng Pilipinas at ibang bansa. Sa aming kapamilya sa PCOM, mga kapuso sa OH at kapatid sa propesyon ng kalusugan. Isang mainit na pagtanggap mula po dito sa amin sa PCOM Quezon City Chapter. Isang taon na din na lumipas mula nang tumama ang COVID-19 sa bansa. Isang pandemya na gumulantang sa sangkatauhan. Ito ay malabangungot na kabanata sa karamihan na pilit bumabangon mula sa matagal na pagkakahimlay. Maraming sayentipiko at dalubhasa ang patuloy na nag-aaral at nagsasaliksik ng kaalaman magpahanggang ngayon upang tuluyang masugpo ang pandemyang naglugmok at humamo sa katinuan ng madla. Sa pagtatapos ng taon 2020, lumabas na ang iba't ibang bakuna panlaban dito sa COVID-19. Ngunit madami pa ring agam-agam ang karamihan sa mga Pilipino. Kung ito na ba ang tutuldok sa pandaigdigang problemang ito? Ngunit sa pagpasok ng taong 2021, umugong ang balita na ang bagong UK variant ng COVID-19 ay nandito na sa bansa. Patuloy na nagpapadagdag sa takot ng mga Pilipino. Kaya po ngayong gabi, sa aming ika-14 na anibersaryo ng PCOM Quezon City Chapter, kami ay lubos na nalulugod dahil kasama po natin ang isa na namang eksperto sa larangan na ito sa ating ika-25 kapihan para talakayin at magbigay linaw sa bagong issue na ito. COVID-19 strikes back, the rise of the UK variant. Once again, happy 14th anniversary sa ating lahat na bumubuo at tumatangkilik sa PCOM Quezon City Chapter. Naway patuloy po kaming makapagbigay ng kaalaman, pag-asa at liwanag, dala ng karimlan ng kasalukuyan. Maraming salamat po at magandang gabi po muli sa inyong lahat.
Maraming salamat, Dr. Catherine Artais Carriaga, our PCOM QC Chapter President. Ladies and gentlemen, my dearest colleagues, to all our beloved guests from all sectors of workforce, we are very honored that napaunlakan tayo ngayong gabi ng ating panauhing pandangal. So to introduce her, our distinguished and honorable guest tonight, may we call our Chapter CME Chairperson, Dr. Maria Cecilia Caiga Dayana.
Maraming salamat, Dr. Aku. Magandang gabi po sa inyong lahat. Our speaker for tonight is Ms. Lei Lanna Dancel. She is currently the OIC-Head for the RITM Molecular Biology Laboratory. She took up her Bachelor of Science degree in the University of the Philippines and has completed her coursework for Master of Science in Public Health, major in Medical Microbiology, Viro Immunology track at the University of the Philippines, Manila. She joined the RITM MBL in 2009 and became its laboratory manager in 2016, focusing primarily in the strengthening of the laboratory's quality management system for molecular diagnostics for MERS coronavirus, Zaire, Ebola virus, and HIV drug resistance genotyping. She is currently spearheading efforts towards establishing the Quality Assurance Program. At present, Ms. Dancel leads the COVID-19 response efforts of the RITM Molecular Biology Laboratory on molecular diagnostic testing of SARS-CoV-2, SARS-CoV-2 whole genome sequencing.
So without further ado, I would like to give you Ma'am Lei Lanna M. Dancel.
Good evening, everyone, and happy anniversary to the group. Thank you so much for inviting me as a speaker for the 25th installment of Kapihang OH. So for this evening, I was asked to present a briefer on the UK variant. But since there are already additional SARS-CoV-2 variants of public health importance, we will be using the term variants of concern for this purpose. So for this evening, we will be looking at VOCs, as they call it, and their general implications, as well as the WHO recommended response for these variants. Next slide, please. So before all that, I would just like to share a brief backgrounder of what our laboratory does and how we are involved in SARS-CoV-2 variant detection and other response activities, pandemic response activities. So the Molecular Biology Laboratory is the first COVID-19 testing laboratory in the country. We serve as the primary testing laboratory for Zaire Ebola virus, MERS coronavirus, and HIV drug resistance genotyping for the country as well. Right now, we provide training, technical assistance, and QA to the Department of Health, subnational laboratories, and other COVID-19 testing labs. And we also serve as a sequencing facility for SARS-CoV-2 and other emerging and re-emerging infectious diseases. So in summary, we do lab network QA, technical assistance. We produce PP panels for the QA program. And we also participate, well, of course, the Philippine Genome Center is currently the primary sequencing facility right now. We are still awaiting our set of reagents to be able to perform full blast the sequencing for SARS coronavirus 2. And we also participate in a lot of COVID-19 related research work. Next slide, please. So for the first part of the discussion, we will be defining a few terms. I'm sure you're all very familiar of the terms that we will be using, but for the benefit of the other members of the group, we will be defining what is a gene. We will be differentiating it from a genome. We will be defining what is a mutation and how do we detect mutations. And then we will be looking at the SARS coronavirus 2 variants and the variants of concern. Then we will be looking at the implications of SARS-CoV-2 VOCs and the response, the recommended response from the World Health Organization against these variants of concern. Next slide, please. So first, what is a gene and a genome, and what can viral genomes tell us? Okay, so next slide, please. So here you have a 3D structure of SARS-CoV-2 displaying its outer and inner proteins. So very prominent po dun sa labas. On the surface, you have the spike protein, the orange protrusions outside of the structure of the virion. And this protein is very important because it plays a role in receptor binding and entry of the virus and into the host cell. And as you know, this is also a major antigen. Okay, and this is a major immunogenic component of the vaccines. Okay, and the other important part of this virus particle would be the single-stranded RNA genome. So there are a lot of viruses, but basically one category would be the single-stranded RNA genome viruses. And the genome of SARS-CoV-2 contains about 30 kilobases, and its genes code for 27 different proteins. Okay, so a lot of the proteins it displays. So the first one is a spike protein. Then you have the nucleocapsid, the envelope, and the membrane protein. And it's encased in a lipid bilayer. Next slide, please. Okay, so inside the virus particle, as we mentioned, you will have the nucleic acid, and the entirety or the entire compilation of genetic material of the virus would be called its genome. And this contains instructions to build an organism. Okay, so we will be talking a lot about sequences and how they change. So essentially, when we are looking at variants, it's important to denote the particular changes in the genome of this particular virus. Okay, so a genome is different from a gene in the sense that genes are relatively parang small compartments of or fragments of the genome. Okay, they are shorter, and they code for a specific protein. So as I've mentioned before, the SARS-CoV-2 virus has 27, or the genome, or it has genes that code for 27 different proteins. And the genome functions in instruction. It serves as an instruction material for the building of these proteins. Next slide, please. So the entire sequence of the genome serves as the blueprint of each particular virus. So it's the ID of each particular virus. Okay, next slide, please. And if you stretch out the genome, it can be simplistically visualized as such. So here you have a simple visualization that if you stretch it out, you can see that it's parang a string of pearls, no, in this illustration. No, and each particular pearl is a base, no, and we know that bases are called ACTG, so there are different kinds of bases. No, so the sequence or yung order or yung pagkakasunod-sunod nitong mga bases na to, that is the sequence that identifies a particular organism, or in this case, a virus. Next slide, please. So what is a mutation and what does it mean when viruses mutate? Next slide, please. So a mutation is any change in the genome sequence. Okay, next slide, please. So the sequence of SARS-CoV-2 genome may change over time as they multiply and spread over the population. So it's very normal, especially for RNA viruses and coronaviruses, in that, as a manner of speaking, that they mutate. Okay, so habang nagsispread po sila sa human population, the sequence may change. So by random mutations. So in order to illustrate, so the first picture depicted by a red virus. So as you can see, the original sequence is AACAAG. Okay, but as the virus is transferred from one person, okay, in this particular person, it acquired a sequence change. Okay, so the sequence became AACAA instead of a G dun sa dulo. And then when it transferred to another person, it can again acquire another random mutation na nagkakaroon ng pagbabago dun sa kanyang sequence. Okay, so mutations in the viral genome may occur over time until they become different enough to be called a variant strain. So the more chances for this virus transmitting from one individual to the next or over several populations, so the more chances that we are giving the virus for mutating. Okay, next slide, please. So viruses constantly mutate, and new variants of a virus are expected to occur over time. Sometimes virus variants may emerge and they can disappear. So in this illustration, so it is depicted, the first on the left illustration, you can see that the original virus in the population is red colored. No? And then in the second, in the middle illustration, you see that a variant emerged depicted by a green virus particle. Okay. However, naka-acquire siya ng mutation na hindi naman nag-survive over the population. So it can become extinct by acquiring mutations that are not beneficial for the virus itself. So parang detrimental siya na mutation. So hindi siya nag-survive over the course of time while it's being transmitted from one person to the next. Next slide, please. So other times, new variants emerge and persist or stay in the population for a longer period of time. So these are viruses which are able to acquire mutations that are beneficial to the virus itself. So it can acquire mutations that allows it to bind better to receptors of human host cells, or pwede ring mutations to allow it to replicate better inside the host cell, or mutations that allow it to have parang mas productive yung or efficient yung pag-transmit niya from one host to the next. So those are beneficial mutations to the virus. So in this case, the mutant strain of the virus may replace the old ones because it has more beneficial mutations. So it's more adapted. So it becomes more adapted to the human population in this case. So there may become a virus which replaces other virus strains. So that is called replacement. So next slide, please. So how do we detect mutations? Next slide, please. So for us to be able to detect mutations, we have to do sequencing. So and for us to detect a variant, so we cannot just sequence a small fragment of the virus. So it's necessary to perform whole genome sequencing, because we have to determine if the mutation or mutations that a particular virus strain acquires makes it different enough to be called a variant. Okay, so there are a lot of different technologies. So matagal na po yung sequencing technologies. Meron na first generation, second generation, third generation, and they would have, it's not just a matter of having different brands of equipment or technology, sequencers. No, it's sequencing chemistries, magkakaiba po yan lahat. So each kind has its own set of advantages and disadvantages, and labs can choose the kind depending on the lab's objectives and limitations. So sa Philippine Genome Center, they have a higher throughput of equipment because they have a different set of objectives than from RIPM. And for us, we have a smaller throughput of sequencing equipment. Next slide, please. But essentially, the goal of sequencing is to really identify the exact order of the bases for the entire genome of the virus. So that's the goal. And SARS coronavirus 2 sequencing in the Philippines is not just parang an effort from Philippine Genome Center and from RIPM. So there are a lot of units that are involved para po magawa po yung whole genome sequencing, because we have to get samples from all over the Philippines, because it's not sufficient to just sequence kunyari, samples from a particular area like NCR, one region. So it's important for us to have representativeness in terms of the sequences that we generate, so that our inferences will be parang pwede tayong makapag-conclude for the whole Philippines. Okay? So it's a joint effort among various pandemic response units, and these are Department of Health Epidemiology Bureau, and of course they are supported by the regional epidemiological surveillance units. So each particular region would have surveillance units in place, and these help us collect the samples and coordinate specimen submission to the sequencing facilities. So that would be RIPM and UP Philippine Genome Center. UP-NIH as well, and of course we have the subnational laboratories and other COVID-19 testing laboratories, which are functioning right now as COVID-19 also testing labs. Okay. Next slide, please. So just to give you a brief overview of the entire effort po, no? So the positive samples, the COVID-19 positive samples from each facility, testing facility, are pooled and they are coordinated to be transported to the sequencing facilities. So in this case, this is RITM, no? And next slide po. And the samples have to be transported in cold chain. It's very important that they are to avoid yung pag-freeze to ng samples, no? Yung parang frozen siya and then matothaw siya, because the frequency of freeze-thaw cycles, or the higher the number of the freeze-thaw cycles, nakakadegrade po siya ng nucleic acid material in the specimen. And it's possible na kapag pangit na yung quality ng specimen, ang nangyayari po is there is nucleic acid material from SARS coronavirus 2, but we will not be able to sequence it because hindi na maganda yung quality, hindi na namin ma-recover sa aming sa course ng pagsisequence. Okay, so here you have an example of what happens in the laboratory, no? So once the specimens arrive at the facility, they are again retested for SARS coronavirus 2 just to confirm if meron pa tayong sufficient nucleic acid material present in the specimen. And once again, of course, since these are already positive samples, it undergoes virus inactivation. I'm sure you've heard of yun, specimen inactivation muna bago mag-extract ng nucleic acid. So ganun po yung ginagawa sa mga testing laboratory. So there's a process, no, na kailangan muna nating isubject sa inactivation, meaning pinapatay yung virus bago natin kunin yung nucleic acid, extract using an extraction kit. Okay. Next slide, please. Yeah. And then we begin the sequencing library preparation, and then after that, we will then load it onto the sequencer for the run. So there are, at RITM, we use a MinION and a MiSeq, Illumina MiSeq. And then, after po noon, we proceed with, next slide please, bioinformatics analysis. In bioinformatics analysis, it's a computerized software, or ang ginagawa po is that the sequencing reads are analyzed, and we are trying to reconstruct the genome because short reads yung napoproduce, and we reconstruct the genome using a reference sequence. So hanggang sa mabuo natin yung genome, yung sequence, genetic material na sequence nung isang ng mga samples na that we are trying to sequence. Next slide, please. So this is an example of how we visualize sequence data. So these are various tools that we use. So these are phylogenetic trees. So we want to understand the relatedness of the sequences. So the more that they are closely grouped together, ibig sabihin mas related or mas similar yung mga sequences nila. And it's also possible to visualize it such that you can spread it across a particular geographic region and visualize kung ano po yung mga lineages or variants na nandoon sa isang particular region. Okay, next slide, please. Again, this is MicroReact. This is an example of a visualization of the sequence data that is being produced. So we want to determine the global pattern of distribution of SARS coronavirus 2 strains. So that's why world map yung nakalagay dyan. But of course, when you're just analyzing the data of the Philippines, it's also possible just to select the strains that we have sequenced for the Philippines. But in analyzing the sequence data for the Philippines, it's always good to parang relative siya to what is happening in other regions, so that we will have a perspective of the strains that are coming in, saan ba ito galing, kanino related yung mga sequences na galing dito sa Pilipinas. And it's also possible to have a temporal distribution, no? So parang pwede mo siyang i-timeline such that we are able to see kung ano yung mga sequences ng SARS coronavirus 2 from, let's say, for example, from Q1 of 2020 hanggang sa kung ano yung data na na-produce mo for your sequencing run. Next slide, please. Yeah, so this is just so on the upper left quadrant, you have the geographic distribution, and on the bottom part, you have the timeline. And on the right part, you have an annotated view. No? So makikita mo dyan yung mga virus strains na sequence and how they are related with one another. So the mutations in the SARS coronavirus 2 genome have been observed globally. So as we know, as we've mentioned before, mutations occur randomly, and it's a natural process for viruses, especially coronaviruses. No, so they constantly evolve, and dominant virus strains may change over time. So yung mga dominant virus strains from the first quarter of 2019, hindi na po ganun yung na-observe natin sa latter part of first quarter of 2020, hindi na ganun yung na-observe natin for the latter quarters, especially ngayong pumasok ang 2021. Next slide, please. Yeah, so this is an example, no? So ito pong ang pinaka is the D614G mutation that was observed in sequences, no? So nakita po natin sa it's a global phenomenon, no? So nagkaroon ng rapid increase in the appearance of this mutation in the sequences that are being generated and reported on a worldwide basis. So it's not just sa Philippines, no? It's all over. So nakita natin yung parang replacement also. So the SARS coronavirus 2 variant containing the D614G mutation quickly spread around the world in mid-2020, and some variant, on the other hand, became extinct. Okay, next slide, please. So let's now look at the variants and what are the variants of concern. So it's not, so in general, variant strains are considered to be of greater concern to the population when first, kung meron siyang increased severity, or kung naka-acquire siya ng property for it to spread rapidly, so mas increased yung transmission, or yung mas infectious siya kasi yun nga, mas mabilis siyang mag-spread. And it can also be a concern if it's no longer detectable in the current diagnostic assays that we have. So that would be, you will be generating false negatives, no? So akala natin negative yung sample, pero yun pala hindi na siya, merong mutation dun sa diagnostic target ng assay mo, such that a sample is declared negative, pero yun pala positive siya. And it can also mean that there might be a change in the response to antibodies. So this is the actual real concern right now. So parang hindi natin sure kung that is the general worry because of the vaccine rollout. Okay, so what are the current SARS coronavirus, next slide. SARS coronavirus 2 variants of concern. So now we have 3. So the first is the UK variant. This is also called the B.1.1.7 lineage, or B117 lineage. So it was first detected in September 2020. So ang sinasabi nila is it was first detected in UK, that's why it became the UK variant, but it's also possible that it arose from somewhere else. But it was because very massive po yung sequencing efforts nila sa UK, doon siya nakita. Okay. So hindi pa rin sure kung saan siya nag-arise na country, kung totoo bang sa UK or from somewhere else. And it was also detected in other countries. So, ang bilis niya ding nag-spread, no? So, greater than 50 countries na. So, to date, ang nire-report ng UK variant, including the Philippines. So, the general concern for this is, so among the criteria natin for saying that a variant is a variant of concern, ang kanyang concern specifically is increased transmissibility. Okay. So the second variant of concern is the South African variant. It was first detected in October 2020, and it was detected in South Africa. And yes, it was also detected in a lot of other countries, but hindi naman over 50, no? So less than 20 countries. And the general concern that they are looking at is increased transmissibility and possible reduction of vaccine effectiveness. For the third variant, we have yung most recently lang siya na-detect is the Brazilian variant, also called P.1. It was first detected in December 2020, and it was both detected in Brazil and Japan. The general concern about this particular variant is that it is associated with increased transmissibility and possible reduction as well in vaccine effectiveness. Next slide, please. So all three variants, ang common na concern is that they spread faster. So associated sila with increased transmissibility. So in the countries that were reporting this particular variants, biglang double or nagkaroon ng rapid increase in the number of cases during the months that the variants were reported. However, laboratory studies are ongoing to determine whether these variant viruses have different biological properties, or if they are able to alter the vaccine efficacy. No, so as of now, there is not enough information to determine if this variant is associated with any change in severity of clinical disease, antibody response, or vaccine efficacy. So ang clear lang is that they spread faster. Okay. And according to WHO, so next slide, please. So they came out with a statement that at this time, most scientists believe that the vaccines that are currently in development, and a couple that have been approved, are able to provide protection against these variant strains, because the vaccines generally elicit a fairly broad immune response. So it's not just a host of antibodies that are being produced. There is also cell-mediated immune responses. So aside from the antibodies themselves, itong vaccines natin, currently in the market, they elicit yung mga T-cell na responses natin. So as of now, wala pa pong napapatunayan na nakaka-reduce siya ng effectivity or efficacy ng mga vaccines. Kumbaga, there's no evidence na hindi tatalab yung mga bakuna dito sa mga variant strains na to, precisely because of there are other immune responses involved. Okay. Next slide, please. So what are the implications of these variants of concerns, and what are the recommended response? Okay, so these variants, which are associated with increased transmissibility, these are not to be dismissed. Kumbaga, meron talagang real danger because they lead to increased number of infections. Okay, so hindi sila dapat ipagpawalang bahala. Okay, so kung dumadami yung number of infections, mas dumadami yung nangangailangan na ma-hospitalize. So, higher hospitalization and possibly death rates because yun na nga, mas marami ang naapektuhan at any given time. So, they put significant pressure on the healthcare system and nakikita naman na natin yung epekto na yun. That's why there's a real clamor for the rollout of the vaccines. Next slide, please. So what is the global strategy for SARS coronavirus 2 variants of concern? So the potential for virus mutation increases with the frequency of human and animal infections. So ang strategy dito would be to limit the virus's opportunity to spread and mutate. So the WHO recommendation is still to strengthen existing disease control activities to reduce transmission. So we want to reduce the transmission to limit the spread and the possible mutation because of the spread. Because as we've mentioned in the previous slides, the more that it spreads, the more chances for mutation and the generation of variants, which right now wala pa siyang association with increased severity, no? So hindi walang ang sinasabi nila is that mas mabilis siyang kumalat. Okay. So the call to reduce the transmission is still includes the following, no? So pag-monitor ng epidemics through epidemiological surveillance and strategic testing. So that is what we are doing right now. And yung pagpapatuloy ng outbreak investigations and contact tracing efforts. Next slide, please. By reducing transmission, we mean the following, no? So yung katulad lang din ng existing natin na health protocols, so avoiding close contact with people suffering from acute respiratory infections. So it's the same frequent hand washing, especially after direct contact with ill people or their environment. So people with symptoms of acute respiratory infection should practice cough etiquette, maintain distance, cover coughs and sneezes with disposable tissues or clothing, and wash hands. And within the healthcare facilities, we have to implement enhanced standard infection prevention and control practices, especially in emergency departments, and of course the general wearing of masks where appropriate and ensuring good ventilation. Next slide, please. So another important strategy or global strategy is to monitor and track how the virus is changing. So another WHO recommendation is, where feasible, increase the routine systematic sequencing of SARS coronavirus 2 to better understand its transmission and to monitor the emergence of variant strains. No? So it's important that in the Philippines, we have a monitoring system kung ano-ano na ba yung nagsisirculate na virus lineages, ano-ano na ba yung nag-generate ng mutations. And you should have a system for tracking or monitoring the emergence of variant strains and how they affect us. No? So this can only be affected through genomic surveillance. Okay. So WHO, at the WHO level, they created the Virus Evolution Working Group. Here in the Philippines, the IATF, there's a task force for variants of concern. So PGC and RITM were activated to perform whole genome sequencing of SARS coronavirus 2, and genomic surveillance is being established right now. So that's what they call it, the biosurveillance. No? So what? Next slide, please. So why do we need to monitor the viral genomes of SARS-CoV-2? Next slide, please. We need to know how this virus is moving. So regular, strategic, and purposive sampling of positives for sequencing can show cases which are related. So malalaman natin kung merong clustering and estimate from where the virus strains came from. So from the visualization tools that I showed you earlier, we will be able to see kung saan mas related. So yung sequences na na-generate dito sa Philippines, and we can make inferences kung saan siya galing. kung saan posible siyang nanggaling itong virus strains na to. So, genomic surveillance significantly aids contact tracing. So, sana magkaroon tayo ng maraming reagents dito sa Philippines to be able to support timely na sequencing of these variant strains or these virus strains. Next slide, please. We also need to know if the virus is becoming more harmful. No? So, right now, this is not the case, but it's important na in place yung mechanism natin to be able to detect kung yung mga strains na nandito sa Philippines or sa neighboring regions natin or neighboring countries are producing or associated with more severe illness. No? So currently, again, just to reiterate, there is no evidence that the UK variant, the South African variant, or the Brazilian variant produces more severe illness than other SARS coronavirus 2 variants. Next slide, please. Okay, so we also need to understand how it is changing, no? Why? Because very dependent ang ating mga response sa pag-determine if a sample is positive or not, because that is the starting point, no? So we need to detect if a person, kahit nag-uumpisa pa lang siya ng kanyang sakit, ay dapat ma-detect na ng mga assays natin. However, this would be impaired if the virus would acquire mutations nga, like I mentioned, doon sa diagnostic target. So as of now, each laboratory in the network, they were able naman to check their particular assays. So dito sa RITM, we immediately contacted our manufacturers, the diagnostic kit manufacturers, to determine to ascertain na hindi apektado ng mutations from or the associated dun sa mga variant strains yung ating mga diagnostic tests. Okay, so all of them are able to detect naman SARS coronavirus 2, including the variants. It's also important to determine if the mutations that the virus acquires ay hindi nakakaapekto sa current na treatments. Okay, pero caution din kasi the UK variant, so it was associated with a person with a chronic illness and got treated with antivirals. So nagkakaroon din kasi ng pressure for the virus to mutate if meron siyang prolonged exposure to, of course, yung antivirals. Malaki siyang pressure to mutate for the viruses. So meron ding dapat caution dun sa mga antiviral, pag-administer ng mga antiviral treatments. So, and then of course, will the SARS coronavirus 2 vaccines work for the variants? So right now, like what we said, wala pa namang evidence, okay, that indicates that the vaccines won't work. And the general recommendation is, the statement from the WHO is that broad naman yung protection na nai-elicit ng mga current na vaccines in the market. So nagsasuggest ito na they will be able to neutralize the variant strains. So all of these things, genomic surveillance can provide essential information to be able to identify or detect itong mga ganitong klaseng changes. Yun. So, ayun. So that ends my presentation po regarding, it's a briefer again for variants of concern. I hope I was able to shed light to some of your queries regarding variants of concern, especially the UK variant. So, maraming salamat po for your time.
Maraming salamat, Ma'am Dancel. Ma'am Ma'am Lei, are we ready for the open forum po? Can we ask questions na po with regards to the para ituloy-tuloy ko na po yung mga clarifications of our topics?
Yes, please po.
Okay. So let me start by this question. Based on the data available, which of these variants is more virulent than the original strain? You have presented as the UK variant, the South African variant, and the Brazilian variant. No? Yun po yung tatlo. Doon po ba sa datos po, sino po kayang pinakamatapang doon?
So as of now, like I mentioned po, no, so wala pong datos na nagsasabi na itong mga any of the variants of concern na na-mention natin, the UK variant, the South African variant, and the Brazilian variant, na nagsasabing mas severe yung illness na produce nila, no. So when we say na mas transmissible siya, ang nakita nila based on the laboratory data from research is that mas madami yung virus particles, okay, na na-generate during the replication of the virus. So, ang nangyayari, so let's say, imagine natin, kada bahing ng isang tao, kada sneeze niya or cough niya, mas maraming virus particles ang pwede niyang ma-transmit. That's why it's more infectious, no? So, with that in mind, I hope po na maging vigilant pa rin tayo at hindi tayo magkaroon ng lax na mindset towards the infection control measures that we are implementing, not just within our workplace. Actually, mas important po yung pag-outside na, diba? Like, kunyari, you're commuting or you're nagaano po kayo outside of your homes and even inside. So, lalo na po yung, syempre, yung mga mas at risk natin na family members. So, yun po. Right now po, wala pa pong data to suggest that.
Okay. With regards to that, ma'am, meron bang sinasabi ring affected population right now na mas apektado of all this strain?
So, the same thing po. So, ang at risk po sa population natin would be those na elderly and also yung may mga comorbidities o yung meron pang underlying na sakit or merong iba pang sakit. So, para siyang more chances of winning to si SARS-CoV-2 kasi nga so mas madami siyang virus particles na na-generate. So, yun po. It's the same the same din po siya na risk for certain members of the population.
Okay. Nandito na rin tayo sa vulnerable side. Is there, what do you call that? Is there a need na papalitan po natin yung ating way of wearing our mask? Meaning to say, is there a need to require na mag-N95 mask na lang than surgical mask or those DIY masks?
As of now po, so every time po na merong mga bagong pangyayari, we always look at what the WHO would recommend. So right now po, there is no advice with regards to the pag-level up po ng ginagamit po natin na masks. Ang important po kasi right now is to stick to the health protocols. So, yung physical distancing coupled with mask wearing and hand washing, yun pa rin po yung WHO recommendation for personal protection and also community na protection, no? So, if we stick to those, then supposedly, hindi malilimit po natin yung transmission. So, ganun pa rin po yung advice as of now.
Okay, thank you for that. Now, how often do we do the sampling or sequencing? Tuwing every month ba yan, ma'am? Or every week? Para malaman kung may bago tayong strain na de-develop.
Opo. So, since si Philippine Genome Center po, meron po siyang mga high throughput, ibig sabihin yung mga equipment, sequencing equipment na mas marami yung samples na kaya nila in one time. So, they are able to sequence 700 plus samples every week. Okay.
Every week,
Opo. Every week po yung pag-sequence niya. So, naghihintay lang din po tayo ng more na reagents. So, katulad po ng lahat ng materials for the response like PPE or PCR kits or nucleic acid extraction kits, worldwide po yung need. That's why sometimes it takes a longer time to purchase them and to be able to source them out, like what's happening with the vaccines, no? Ganun din po yung current situation. So, yeah, so on a weekly basis po, si Philippine Genome Center, as I've mentioned before, yung sa RITM naman, mas makaunti yung number of samples na kaya ng ating equipment. So, hindi as much as 700 per week yung kakayahan. So, yun po.
Okay, I see. So, nandun na rin tayo sa mga variants and genomic sequence. So, aside from the Bondok Mountain Province B117 variant cases, were there any other community transmissions documented based on genomic surveillance?
So, as of now po, yung pong mga, ang nakadetect po ng UK variant, no, within our communities, so mga samples from CAR region, no, I see Philippine Genome Center, and they would post their bulletins in their website, the PGC website. Okay, so nandun po yung mga latest din po na mga but of course, as since nandito na si UK variant, and we know that mas mabilis siyang kumalat. Okay? So meron talagang possibility na magkaroon ng mga community transmission, but the hope is, since alam na natin ito, that we will be able to control this by implementing properly our health protocols. Na katulad nga lang din po nung ibang na-mention kanina, which is yung important talaga na mag-stick pa rin tayo sa mask wearing, physical distancing. Kung masama ang pakiramdam, wag lumabas. Yung mga ganong bagay. Pag-iwas po sa mass gathering, kasi parang habang tumatakbo po yung panahon, nakikita natin parang medyo nagiging lax yung publiko. So, it would really help if, ang tawag dito, hindi po tayo maging kampante.
Totoo naman po yan, ma'am. Now, para hindi sila makampante, kumbaga, ano na po ba ang status ng ating na-mention na mga variants, and is there other variants identified aside from these three?
Yes. So, actually, dito lang sa Philippines po, marami na po tayong mga variant strains na na-detect. However, like I said, we are just highlighting those of concern because some variants, they are just similar in terms of biological property as with the original strain. No? So, walang pagbabago doon sa kanilang clinical manifestation, or let's say yung transmissibility nila, it's the same. No? So, marami na tayong nakitang strains from those, of course, from the strains, the original lineage from China. Meron ding mga European lineage. Merong mga Hong Kong lineage, Spanish lineage. Marami na po kasi sobrang virus lineages itong si SARS-CoV-2. However, what we are watching out for are the introduction of these variants of concern. So, out of the 3 variants of concern, as of now, si UK variant pa lang po yung na-detect, specifically ng PGC, na nandito po sa Pilipinas.
I see. Now, ang ginagawa natin ngayon ay nag-RT-PCR, which is the gold standard. Now, there's a new way of testing. It's called the saliva RT-PCR. Is this a reliable method on detecting the SARS-CoV-2 variants? All variants, to be exact.
As of now po kasi, meron ongoing po yung mga studies, no? So there are several groups studying the saliva as a specimen. So hindi po kasi basta-basta tayong pwedeng mag-shift without really evaluating if it's equivalent in terms of maging specimen for detection of the SARS-CoV-2 viral RNA. But it's a promising specimen. That's what I can tell you right now. But hindi ko lang po pwedeng pangunahan yung mga resulta po ng mga ongoing natin na studies. So hopefully po, pag dumating na yung mga resulta, we'll be able to share, of course, the results of the study via the Department of Health. But it's a promising specimen. So less invasive siya. They call it less invasive because hindi na
Yan sa ano na po,
Yes,
nadulo
hindi
ng ano talaga.
Opo. So, yon, maganda po siya sanang alternative. We're just waiting for the results po.
Okay. There are just few questions na lang po, no, ma'am? So, how long will the genomic sequencing be made available to the patient? Is it important to know the genomic sequence? Maapektuhan ba ang treatment natin, ma'am?
So, wala pong pinagkaib right now with the variants na nadedetect po. Wala pong siyang pinagkaiba in terms of hindi po maaapektuhan ng patient management.
Okay.
Okay? So, wala pong special na gamot na maibibigay kapag nalaman ng isang pasyente kung ano yung strain niya. Yes. So, ang benefit po talaga ng genomic sequencing is for the population level. Okay. So we are able to detect kung saan siya galing, kung ano yung parang it has to be an aggregate data. It's not really for individual na benefit but on the population level. So ang isa pa pong call ni World Health Organization is for the sequencing facilities to upload the sequences kaagad sa mga public repositories so that other researchers are able to analyze. Kasi yan pong mga sequences na sinasubmit natin sa public repositories, they are being used for vaccine development. So let's say, kunyari hindi tayo nagsisequence dito sa Philippines, no?
Okay.
Hindi represented yung virus strains na nandito sa pag-formulate ng mga bakuna or even ng mga treatment or sa diagnostics. So, if hindi natin alam what the sequences of the virus here, yes, hindi tayo represented. So, it's important na makapag-participate tayo sa level po na yun.
Okay. That's good to know po. Ano? Now, when it comes to participating and then the testing nga naman, what is your personal take sa rapid antigen test? Madedetect din ba niya yan na initial screening for the new variants?
So sa rapid antigen test po, so hindi naman, alam naman po nating lahat na hindi magkaiba po yung sensitivity. Okay?
True.
Ibig sabihin ng sensitivity, ang impact kasi when you say that an assay or a test is more sensitive, ang benefit po niyan is kapag mababa yung viral load mo, kaya pa rin niyang ma-detect. So ang implication po nito is that if a person is nag-uumpisa pa lang ang kanyang infection, so you would expect na kaunti pa lang yung viral load or yung virus particles na nandun sa sample niya. So ang test ng isang assay is if madidetect niya yung sobrang konting virus particles na present doon. And mas sensitive pa rin po right now ang RT-PCR testing compared sa antigen testing. So ang ibig sabihin nito, there's a possibility na dun sa antigen testing, hindi niya na ma-detect yung mga either paumpisa pa lang or patapos na. So, yun po yung difference.
Okay. Now, how long will the genomic sequencing be available to the patient as it was sequenced? Kumbaga, once na na-detect na meron siyang SARS-CoV-2, na kailan po madedetect naman ang genomic sequence niya?
Opo. So like I said po, hindi po siya, so ang nangyayari po kasi diba, magpapatest si patient, let's say RT-PCR, so it will be shared sa kanya immediately, no? Pero like I said, yung sa genomic sequencing po, on a weekly basis siya. So alam na ni pasyente na let's say positive siya.
Yes.
So, na-manage na siya ng kanyang doktor and na-quarantine na siya and also nakapag-contact tracing na sa pamilya niya. So, ito pong benefit again ng genomic surveillance or genomic sequencing is at the population level. It's not on the
Population.
individual level. It's
Thank
the
you.
information is not for patient management po. So, yun po yung ano niya.
Ay, salamat, ma'am. So, ibig ba sabihin nito, ma'am, for the educational purpose, hindi naman maaapektuhan yung nagawa na nating mga guidelines and protocol, like saka pa lang ire-report sa LESU kung nalaman na natin ang genomic sequence, ganon, no? Yung ibig ko sabihin is second and third generation close contact tracing, mga ganon po. So,
Opo.
as always, pareho pa din ang pattern of
Yes po.
report.
Yes po. So the moment na mag-positive yung sample, of course, ima-manage na po siya. So dapat yung na-initiate na yung mga contact tracing and quarantine measures. And so hindi na dapat hintayin yung genomic sequencing result.
Yes.
Okay.
Opo. Thank you.
Opo.
Thank you for clarifying that. So hindi na
Opo.
kailangan antayin pa
Hindi
yung
po.
genomic sequence. Yes.
Hindi po.
Ano
Opo.
naman, once positive sa RT-PCR, we do
Yes
the
po.
treatment at once. Now, last question, ma'am, para po. How do you determine which sample is needed for genomic surveillance? And is there a single test run to be able to detect the multiple types of SARS-CoV-2 in one specimen?
Okay po. Maganda yung question. Gusto ko yung question
Thank
po
you.
na yun. So,
Paano yung ending?
Opo. So, yung pong ang criteria po for samples for genomic sequencing. So, lahat po ng COVID-19 testing laboratories, binigyan po yan ng guidelines ni Department of Health. So, lahat po ng positive samples na maganda yung CT value. Okay po. So, meron po kaming sa lahat po ng testing labs na RT-PCR yung ginagamit or majority of them, meron pong na-generate na CT value. It's a value ng run, no? So, yun po yung nagiging criteria namin para malaman namin kung pasok ba siya, pwede ba siya for sequencing or hindi. Because masyadong mahal ang reagents for sequencing at kaunti lang yung, kumbaga parang limited yung volume of or availability of the sequencing reagents. That's why we have to prioritize it for samples na maganda pa yung quality or yung concentration ng nucleic acid material. Kasi kung kaunti na or hindi na masyadong maganda for sequencing, sayang lang yung isang sequencing run. Sobrang mahal po ng sequencing. So malaking investment po sa isang bansa to engage in genomic surveillance. That's why we are there's a criteria, no, to ensure na bawat sample na isasalang natin for sequencing would have a high chance na makaproduce siya ng sequence data. Now, yun naman pong question about, is there a single parang test, no?
Yes.
To determine if a sample has parang multiple na variants. So, meron ang tinatawag po namin doon is metagenomic analysis. And this is, yes. So, you can also parang sequencing based din itong method na ito, no? So we can do next generation sequencing and run a metagenomic analysis or a bioinformatic analysis na pipeline to be able to sequence lahat ng pathogens for that particular sample, no? So however, this is more expensive, more costly. So kailangan nating i-limit kung ano yung objective, no? So kung right now ang objective natin for sequencing is to determine if nandito na ba yung mga variants of concern, and also to monitor kung ano na yung mga lineages na nagsisirculate sa Philippines, to have an insight kung saan sila galing, no? Kasi itong mga insights na ito, pwede natin siyang gamitin para makapaglagay tayo ng mga measures like kunyari border control, yung mga ganon, kung saan tayo dapat ipaigtingin yung pag-iingat. So ganon po.
Meron ba kayong mga pinipiling sample, person na, ito, i-genomic natin ito, na strain niya or something? May mga ganun po ba? Ganun ba ang basis nito na pipiliin? Or lahat naman nakukunan na lang ng random sample?
So, mas importante pong criteria is yung representativeness sa bawat rehiyon ng Pilipinas, no? Ang gusto sana natin is that makakuha tayo from all regions of the Philippines, no? And also, so makukuha tayo ng mga representative ng mga positives po doon, napasok doon sa
Okay.
criteria in terms of, yun nga, yung quality ng specimen, concentration, and also yung volume ng natitirang specimen. Kasi baka maganda nga siya, pero hindi na sufficient yung volume ng sample para makapag-sequence. So, yun, right now, yun lang po yung criteria.
Oh, thank you so much, Ma'am Dancel. Very informative po itong ating talakayan. Now, marami pang mga tanong. However, where can they reach you para pwede kanilang i-email or ano po about their concerns with regards to our topic tonight?
Opo. So, i-message ko na lang po sa chat box so that
Oh, thank you.
opo, ilalagay ko na lang po doon for the benefit of everyone po.
Our chapter Vice President, Dr. Jeremias Martin Florante.
Maraming salamat, Dr. Aku. It has been another fruitful kapihan for all of us. I remember the lecture of one of my professors in microbiology that among the microorganisms, the virus is prone to mutation because of its structure. Hence, only a matter of time before a variant of SARS-CoV, the causative agent of COVID-19, will become available. Our only hope is that it will not be so virulent and the mode of transmission will not change. Else, we will be faced with another challenge. In behalf of the Quezon City chapter of the Philippine College of Occupational Medicine, allow me to express our heartfelt gratitude to Ms. Lei Lanna Dancel at the Research Institute of Tropical Medicine for imparting to us her knowledge on new SARS-CoV-2 variant strains. Hindi lang pala siya isang variant ang mayroon tayo, kundi tatlo at maaaring sumobra pa. We hope that the information we have learned tonight will help us in the evaluation of our existing programs and activities on the prevention and control of COVID-19 in the workplace. Allow me to express our appreciation also to the past presidents and to the national officers and board members of the Philippine College of Occupational Medicine, headed by Dr. Phil Pangilinan, who are here with us tonight. Likewise, to all the chapter officers, board members, and the membership of the Quezon City chapter, and to the general membership of the college. Thank you very much. Without your support and cooperation, we will not reach this far. To my fellow advocates in the field of occupational safety and health, occupational health physicians and nurses, occupational medicine specialists, and safety officers, we recognize your presence in all our kapihans. To all our sponsors who are here tonight, a big thank you. And speaking of sponsors, allow me to plug our partnership again with ISI Pharmaceutical Inc. We are inviting all occupational health physicians, occupational medicine specialists, and all physicians from other specialties who are here with us to join on the 20th of February this year at 5 o'clock in the afternoon to our virtual PH Pulse. Registration links will be posted in our official social media pages for your perusal, and we will be sending notices to via email. This kapihan will not be possible also if not for the active contribution of those in the PCOM Quezon City chapter webinar team. Guys, thank you. Congratulations and kudos. After this kapihan, we will be having 2 premieres showing via our official YouTube channel. The second series of OH in a Flash, and the 24th Kapihang OH that discussed on telecommuting in the midst of the pandemic. So do not forget to like, share, subscribe to our official YouTube channel, and click the notification bell so you will be updated of our new videos. Make sure also to follow, like, and share our Facebook, Twitter, and Instagram accounts for more information on occupational safety and health. If you have any suggestions for our next kapihan topic, please email us at pcomquezoncitychapter@gmail.com Always remember that in PCOM Quezon City Chapter, we value your safety and health. Hashtag PCOM QCC Cares. Muli, maraming salamat po at isang ligtas at malusog na gabi sa inyong lahat.
Okay, thank you so much, Dr. Jing. Sa lahat po, maraming salamat sa pagdalo. Sa susunod po sa 26th Kapihang OH and our collab webinar with High SI this February 2021. Okay? Ako po ulit si Dr. Margo Venuscu, ang iyong moderator for tonight. See you again. Parating nagpapaalala sa inyo sa Quezon City Chapter, ang inyong kaligtasan at kalusugan ay mahalaga po sa lahat sa amin. Hashtag PCOM QCC Cares. Good evening.